COUR. My name is Hannah Kelehear.
And I'm Jacob Parrish.
And we studied the anti-predator behaviors of prey species when exposed to pheromones from the Carolina wolf spider. So in our research over the past few semesters, we focused on pheromones, which are chemical cues released by predators that act much like a footprint to potential prey. Many prey species will detect these pheromones and respond in a few different ways. Additionally, injured conspecific prey will also release alarm cues. When injured or if they come in contact with, say, a much bigger prey, that will trigger these anti-predator behaviors in related prey as well. Additional chemical cues and digestive byproducts of a predator also act as powerful signals that alert to a potential threat. In our study specifically, we examined the behavior of predator-naive house crickets in response to the presence of a mix of chemical cues left by Carolina wolf spiders. We hypothesized that cricket prey would exhibit more pronounced anti-predator behaviors such as increased periods of immobility and would travel less distance when exposed to a mix of predatory chemical cues.
For this study, we purchased adult crickets and collected Carolina wolf spiders from local habitats. We designed 3 different species that would expose cricket prey to a mix of predatory chemical cues for this investigation. In the first treatment, crickets were placed on filter paper that previously held a predatory wolf spider that was not offered cricket prey. This cricket should only detect pheromones left behind by the starved spiders. In the second treatment, crickets were placed on filter paper that previously held a predatory wolf spider that was offered and ingested house cricket prey. In theory, this cricket would detect pheromones, alarm cues, and digestive byproducts. Our third treatment was the control. In this third treatment, crickets were placed on a clean filter paper that was not exposed to predatory wolf spiders. Adult spiders were randomly introduced into round PVC chambers for each treatment that were lined with filter paper. An isolation tube placed in the middle of the chamber prevented spiders from contacting the filter paper and depositing cues in the particular area. The spiders remained in these treatments overnight and were later released unharmed. At the start of each behavioral trial, predator-naive crickets were inserted into isolation tubes positioned in the predator-free circle of the filter paper that contained no chemical cues. The nocturnal crickets were left to acclimate under a dim red light bulb for 10 minutes. After the acclimation period, the isolation tubes were removed and the crickets moved freely throughout the chamber and were recorded by a GoPro camera. The recorded MOV file was then converted to an MP4, and we used the behavioral tracking software program, ToxTrack. This program allowed us to view various anti-predator behaviors exhibited by the cricket prey, including total distance traveled, the mobility and mobility rate, and the average speed of movement. As hypothesized, cricket prey exposed to a mix of chemical cues travel less distance than those of other treatments. Unfortunately, ToxTrack was unable to track relatively few crickets across the full 5-minute duration, so our sample size is limited and our results are questionable. Our analysis found no statistical difference between the mobility rate of cricket prey across the treatments. Again, the increased sample size of these trials allowed us to also statistically compare the average speed of crickets exposed to each treatment. Although crickets in the fed treatment appeared to travel at a faster average speed, these results were not statistically significant as well.
So our hypothesis, again, predicted that cricket prey exposed to a mix of predatory chemical cues would travel less distance than those from other treatments, which was based on the Storm and Lima paper from 2008. Although our data appear to support this hypothesis for crickets in the fed treatment, we're limited by the small sample size and lack of statistical analysis. Unlike Storm and Lima, we found no statistical evidence that cricket prey exposed to multiple chemical cues exhibited greater periods of immobility. However, our mixture of chemical cues differed from those in their study, such as alarm cues, digestive byproducts, mass. There were a few different ones. We also did not observe the reduction in average speed that Storm and Lima observed when cricket prey were exposed to mixture chemical cues. Interestingly enough, it appeared that crickets actually traveled at faster average speeds, although less distance. And again, these results were not significant. The majority of our data remains unanalyzed, and we hope to use additional tracking software programs to increase the confidence in our findings. Unfortunately, because we had to use the field crickets, we did have to use dimly lit red light trials, which ToxTrack struggled to pick up. Moving forward, we plan on using local species of diurnal prey, such as native grasshoppers, which can be filmed in fully lit settings. This expansion of data and trials will allow us to have a more confident result with mobility and average speed going forward. Thank you very much.